Hello everyone, I'm Marion Danjon and I'm a conservator specialized in natural history collection and particularly in botanical wet collections. I'm part of the LICOR project with two colleagues from the Haute École Arc in Neuchâtel, Switzerland. I'm glad to be here to present you some results of our projects, Fluidis and LICOR, in particular concerning the issue of plants' discoloration. The idea for the project came from the observation carried out on the collection of the Botanical Museum of the University of Zurich. Some plants continue to release color in the fluid even after years, and in some cases even after complete replacement of the preservation fluid. Here you can see a Rafflesia plant before and after restoration in 2016. The last picture shows its current state. So, this plant, like other in the collection, is going on releasing color. Those specimens cannot be exposed due to this lack of visibility. Therefore, we started to wonder what can be done to block this phenomenon. So, let's investigate. So, we prepared replicas of botanical specimens in different kinds of fluids used both as fixation and preservation. We monitored them using different tools. The first method is optical examination and photography with fixed parameters such as light, exposure time, and optical aperture. The second method is colorimetric measurements. The colorimetric measurements allowed us to characterize the color change between the beginning of the taste phase and the end. The third method is UV visible spectroscopy. This method is used for detecting which dyes leaked in the fluid. We choose to use dry walnuts because they are ideal for this kind of test. In fact, walnuts were used in the past for ink or dye. In colorimetry, the delta E computes the color change as the difference between the clear fluid at time 0 and the tainted one. Like you can see on the first colorimetric graph, the walnuts first release a lot of dye after stabilizing. Visually, the change of color is very important. What we can say about these results is formaldehyde preserved or fixed samples show faster and more intense leaking of dye. Alcohol preserved samples leaked more progressively. Glycerol preserved samples were the last to leak color. FAA fixed sample in all preservation fluids show slower leakage than formaldehyde fixed ones. On the UV-Vis graph, you can see the different spectra of our solutions. It can be attributed to tannins. Tannins appears on the spectra between 220 nanometers and 400 nanometers, as it can be observed in the reference graphs for the tannins presented on the right. The darker the fluid is, the more intense the UV-Vis absorption is. You can see on these pictures which spectra corresponding to which solutions and also see the different colors. Some results for chili peppers. The release of color is more progressive. The color is going up to a really dark orange. However, after a certain time, the color of the liquids fades and the specimen is whitening. Alcohol preserved samples show a discoloration of the specimen, then a degradation of the dye in the fluid that assumes a lighter color. Formaldehyde and glycerol are grouped together in another color range. In both cases, there is less releasable of color in the liquid. Formaldehyde preserved specimens show a slower discoloration. The specimen became hard and floated. Glycerol preserved specimens retained their color but had shrunk and float. To conclude, a conservation protocol should be chosen carefully depending on which part of the plant has to be preserved, for example, root, flower, fruit, leaf. In addition to the change of color, we have observed a change of texture, whitening, darkening, and shrinking of the specimens. Currently, we are investigating methods for the fixation of the green plants thanks to the work of a master student of our institute. The LICOR project is ongoing and we try to propose conservation solutions and adapting procedures such as fixing the color, preserving volumes, dealing with sealing and air tightness for the different uses of these collections in museums and for research purposes. The team of LICOR project, thank you for your attention.